Hey, I'm Meg Graham. I'm your technologically challenged instructor here, and I've got to get the pen going, so give me just a second. I can't talk and work at the same time. I'm not that coordinated. All right, let's go. Today we're going to talk about cellular respiration. Don't confuse cellular respiration with mechanical respiration. Mechanical respiration is the process of exchanging air between your lungs and the atmosphere. It's breathing in, breathing out, inhaling, exhaling, taking in oxygen, exhaling carbon dioxide. We do all that, though, so that we can have cellular respiration. So that's the whole point of why we breathe. Well, cellular respiration is the process of converting energy that's stored in the food we eat, mainly in carbohydrates or sugars, specifically glucose. And we're going to take that glucose and we're going to turn it into ATP. That's the form of energy that cells can use. So this process requires oxygen, and it gives off CO2. So there is some gas exchange going on at the cellular level, and that's why you get the name cellular respiration. It's not really cell breathing, but there's a gas exchange going on, and that's why they call it cell respiration. If you look at cellular respiration, the whole purpose of this is to produce ATP. Well, ATP is what we call the energy currency of the cell. Currency is money. Think about, like, if you want to go down to the machine downstairs and get a Snickers candy bar. If you have a $1 bill or a $5 bill, you've got plenty of money to buy a $1 candy bar, but your machine only takes quarters. So even though you have the money, you can't buy your Snickers because you don't have quarters. So what you have to do is you have to find a change machine, and that you stick in your $1 bill or your $5 bill and you get quarters back out of it. Then you can take your quarters over to the other machine and you can put them in and you can get your candy bar. Well, in the cell, cellular respiration kind of works as that change machine. It changes energy that's stored up in carbohydrates and it converts it to energy in the form of ATP so our cell can use that. Well, let's look at ATP and see what it is. ATP is adenosine triphosphate. It has a base, adenine, a sugar, ribose, and then 3 phosphates. You see that tri right there? That means 3 phosphate molecules. 1, 2, 3. Well, each one of these phosphate molecules has a negative charge on it. And these negative charges sitting next to each other makes for unhappy little molecules here because negative charges repel each other. So this creates very unstable bonds here between these molecules, and those bonds are just literally bursting, just kind of waiting to blow their energy to somebody else. So when you move a phosphate molecule, or if you break off a phosphate molecule here, that transfers that energy to some other molecule. So when you transfer a phosphate molecule, we call that phosphorylation. So when we move a molecule of phosphate here, that transfers our energy, and we call that phosphorylation. There's always going to be a release of energy with this phosphorylation. This process is how we make ATP, and also how we use ATP. ATP is kind of considered a renewable resource. You can take ATP and you can break it down and use that phosphate for some cellular process, but then later on you can take what's left, this molecule right here, which is adenosine diphosphate. So di tells you that it only has 2 phosphate molecules left on it. And you can take a phosphate and add it back to that molecule and make ATP. So this arrow right here tells you that this equation can go both ways. You take ATP, you use it for some process, and that leaves you with ADP after you pull off that phosphate molecule. Well, then you can take another phosphate molecule for somewhere else, add it back onto that ADP, and make ATP. So it's a real renewable resource, and it's just very efficient for the cell to use it this way. So just remember, the whole goal here that we're trying to do in cellular respiration is we're trying to produce lots of ATP. That's the whole point. Well, we can do it in 2 ways. The first way here is called substrate level phosphorylation. And this is where we do a direct transfer of a phosphate molecule from a substrate to make an ATP. So it's what we were just talking about. So you start out with an adenosine diphosphate, or ADP, which means it has 2 phosphates. You add a phosphate to it, and then that gives you ATP, or adenosine triphosphate. Now, this process is not real efficient, and there's not a whole lot of ATP made in this way. We do make a little bit this way, but this is not how we get the bulk of ATP from this cellular respiration process. So we're just going to put that there's very little ATP made in this way, because it's not the big part of how we do it. What we really do is use this process called oxidative phosphorylation. This is a mass production of ATP using energy that we've stored up in these sugar molecules, and we can make lots of ATP. And we send these sugar molecules, or pieces of the sugar molecules, through something called an electron transport chain. And we'll get into that in just a little bit. But basically, you have what's called an indirect transfer of the phosphate molecule. So you're not just directly taking a phosphate and adding it onto ATP. You're going to transfer it and make ATP that way. Notice here that it's called oxidative phosphorylation. Well, oxidative refers to the fact that it requires oxygen. And so this process does require oxygen. It's really complicated, and I'm going to try to make it simple. I'm going to do my best, but it may come out more confusing. But you know when you log on to Facebook and somebody's status goes, "It's complicated"? Well, this is complicated. We don't need to know why Bob broke up with Sally. We don't need to know who he cheated with and what the whole details are. But what we need to know is that Bob broke up with Sally. So in this case, we need to know we're making ATP, and that's the whole goal. So let's see how we go about making all this ATP. There's basically 3 steps involved in cellular respiration. The first process is called glycolysis, and that's where you just break your initial glucose molecule up into some smaller molecules. And remember, we're always trying to take the energy from these molecules as we break them up, and we're going to use that energy and put it into ATP so the cell can use it. Then you have this little intermediate step right here, which you have to do so that you can enter the second step of this process called the citric acid cycle. So you're going to take your products from glycolysis and you're going to do a little intermediate step on them. Then you're going to enter this cycle, and you're going to extract more energy from them, and then you're going to have some products left over from this cycle that you're going to put through the final part here, which is the electron transport chain, and you're going to make even more energy from that in the form of ATP. So our whole goal here is to take a molecule of glucose, and we're going to turn it into ATP, lots of ATP for the cell. Okay, let's look first at glycolysis. If you look at the name, glycolysis, lysis right here tells you break apart. Well, what are we breaking apart? We're breaking apart this glyco, which refers to glucose. So all we're doing is we're breaking apart glucose in order to release the energy stored in glucose to make ATP. That's the whole point of what we're doing. Basically, this first step of glycolysis, we're just going to take glucose, we're going to break it into 2 molecules. So we take this 6-carbon glucose molecule, we break it in half into 2 3-carbon molecules, which we call pyruvate. So now we have gone from glucose which is a 6-carbon molecule, to 2 3-carbon molecules, and each one of these is called pyruvate. Okay? This whole process does not require oxygen, so it's anaerobic. That means it doesn't require oxygen. It occurs in the cytoplasm, just out in the cytosol. It doesn't require any special organelles, so it's occurring in all of your cells. There's no anything fancy. There's nothing fancy about this. It's just a basic process where we're breaking apart glucose. But in doing that, you're going to get the obvious. You make your 2 3-carbon molecules of pyruvate, but you're also going to get a little bit of ATP. Now, you use a little bit of ATP in order to do it, but you're going to make a little bit of ATP as well. You also are going to create 2 molecules of NADH. And you're going, what the heck is that? I've written down here, and it's an electron carrier. Well, let's look at that for just a second and see what NADH is. It stands for nicotinamide adenine dinucleotide. And basically, this molecule picks up hydrogens and it picks up electrons. And you're going, so what? Well, electrons are energy carriers. So that, if your molecule picks up an electron, it's picking up energy. So now we have our energy or part of our energy that when we broke down glucose is going to be now stored in this NADH. And we're going to take that energy later on in this whole pathway of cellular respiration, and we're going to convert that to ATP. But for right now, we've stored our energy in this NADH molecule. So let's move on and see what we go to next. All right, we broke our glucose down and we came up with our little pyruvate molecules. And I said we had to do an intermediate step. Well, pyruvate is a 3-carbon molecule, and we have to convert that into a 2-carbon molecule called acetyl-CoA. And the reason we need to do that is because that's the molecule that's going to enter our second step of cellular respiration, which is the citric acid cycle. Well, when we do this, when we go from a 3-carbon molecule to a 2-carbon molecule of acetyl-CoA, we're going to make more NADH. So again, we've stored some more energy right there. Well, now we're going to take this acetyl-CoA and we're going to send that through the citric acid cycle. And so that's our second step, so let's go and see what we're going to do there. First of all, remember, we're making ATP. That's the whole goal of this. So we're going to take the energy that's been stored in acetyl-CoA that we've broken down from glucose, and we're going to make ATP. That's the first thing we're going to do. This is considered an aerobic process, but it doesn't really require oxygen. Just know that it's kind of like a gateway to an aerobic process. So it kind of gets thrown in as being aerobic, but it technically does not require oxygen. It occurs in the mitochondria. Now that means you've got to have a special organelle in order to get this accomplished, and along with that special organelle, you're going to have special enzymes. So this can't just happen anywhere. It has to be inside the mitochondria. And then lastly, look at the name, citric acid cycle. It's a cycle. Well, if you have a cycle, that means that you're going to start and end with the same molecule. And that's exactly what happens here. So in a nutshell, I'm going to run through this cycle in 2 seconds and you can see if you can follow me. You start with acetyl-CoA and you're going to put that into your cycle. Well, remember, it's a cycle, so it's just basically going around in a circle here and doing a bunch of reactions. And we're just trying to extract energy as we go around this cycle. Well, we're going to pull it out in the form of NADH. We're going to pull it out a little bit later down here and some more NADH. And then we're actually going to make some ATP. Woohoo, our first ATP here. We're going to make that through substrate level phosphorylation, which again is not real efficient. We're not going to make a lot, but that's one thing that we want. Then we're going to keep going around the cycle, and we're going to make some FADH2, which I'll talk about in a second. And then we're going to make some more NADH. So we take this acetyl-CoA, we enter it into this cycle, and then we're left with all of these molecules right here. Well, as we go along through this cycle, we're going to be throwing off CO2. Remember, you've got carbon molecules here that you have to get rid of. So each time we're getting some energy out of here, we're pretty much going to be throwing off a molecule of CO2. And remember, CO2 is a waste product, so that's why we have to exhale that CO2 and get it out of our bodies. So we take it from the cells into our lungs, and then we exhale it to get it out of our bodies. So that's why we get that cellular respiration name. But anyway, at the end of the citric acid cycle, you've made some ATP, then you've made these NADH molecules that are storing energy, and you've also made some FADH2s. Well, that stands for flavine adenine dinucleotide. It's a similar molecule to NADH in that it is an electron carrier, but it doesn't store quite as much energy. But we'll see, we'll get some ATP out of it nonetheless. So this is a good thing that we've made. All right, well, at the end of this cycle, now we've got our ATPs and we've got these energy storing molecules. So we've got to take those molecules and try to make some more ATP. So that leads us to our last step, or the third step of the cellular respiration process, which is the electron transport series. Basically, this step is going to occur in the mitochondria as well, and it is aerobic, which means it does require oxygen. So you have to have oxygen for this to happen. Basically, you've got some special enzymes in the mitochondria that you need as well. This is called a cascade of oxidation reduction reactions. And you're going, what the heck is a cascade of oxidation reduction reactions? Well, don't get wigged out. A cascade is nothing more than a waterfall. So you're just breaking this one big reaction down into a series of little bitty reactions. Think about like a TVA dam. You've got all this water backed up behind the dam. Well, you don't want to just blow open the dam and let all that water rush through, that's going to be destructive energy. It's not going to be used for anything. It's just going to mow down whatever that water hits. But if you take that water that's backed up behind the dam and you put it through little bitty turbines, you can take that energy in small amounts and you can use it for useful purposes. In a TVA dam situation, it would be for electricity. Well, in the case of the cell, we take that energy that's stored in the FADH2 and the NADH molecules, and we're going to break it down into small steps, and we're going to use that energy to phosphorylate or transfer a phosphate molecule to ADP and make some more ATP. So it says we're using oxidation reduction reactions. Well, what do I want you to know about that? Basically, in oxidation reduction reactions, it's just where you're transferring electrons from one molecule to another. And I don't want you to get hung up on it, but just know that when you're transferring electrons, you're transferring energy and you're releasing that energy. So those oxidation reduction reactions are going to be releasing energy, and that's why they're important. And we're going to do this in small steps or cascades of reactions. Okay, well, after you do all this, basically you're going to take that energy and you're going to use it to take ADP and add a phosphate and make ATP. And you do this, it's kind of like a cellular machinery that does all this. It's an indirect transfer of this molecule. You're using the energy to kind of push those phosphates on. And this is a very efficient process. It produces lots of ATP, and as it turns out, if you take one molecule of NADH, that will actually make 3 molecules of ATP. If you take one molecule of FADH, that will give you 2 molecules of ATP. So this is a real efficient process, and we can make lots of energy this way. So if I haven't totally confused you now, let me try to sum it up and see if we can make it make a little bit of sense. Remember, with the cellular respiration, our whole goal is to make ATP from one molecule of glucose. So we're going to take all that energy that's stored in the glucose and we're going to turn it into ATP, which is the currency that the cell can use. So if you start with our first step, which is glycolysis, you start with glucose, you go to those 2 molecules of pyruvate, and each one of these pyruvates, when you do that process, that's going to release some energy, so you're going to get some ATP and some NADH in the process. But you still have your 2 molecules of pyruvate. So at the end of glycolysis, that's what you have. 2 molecules of pyruvate, a little bit of ATP, and some NADH, which is got some stored energy in it that we're going to get later on. All right, then we go to this little intermediate step where we convert the pyruvate molecules into acetyl-CoA. And when we do that, we're going to release some more energy and stick it in the form of NADH. So now we've got a little bit more NADH, and we can get some energy from that later on. Well, then we go to our second process here, which is the citric acid cycle, and we take that acetyl-CoA and we're going to run it through that citric acid cycle. And as we go around that cycle, we're going to pull out all kinds of energy. We're going to pull out 1 molecule of ATP, we're going to pull out 3 molecules of NADH, and we're going to pull out 1 molecule of FADH2. So at the end of the citric acid cycle, this is what you have left. You have NADHs, FADH2s, and then some ATPs. Everything else about your glucose molecule is gone. So now we take these final molecules here, these NADHs and FADH2s, and we're going to run them through the last step of the cellular respiration process, which is the electron transport series. And that is going to yield, for each NADH, you're going to get 3 ATPs, and for each FADH2, you're going to get 2 ATPs. And that's what you're going to end up at the end of all this. So you start with 1 molecule of glucose, and at the end, you should have 38 molecules of ATPs. Now, I don't want you to learn how to count all those up. That's not my goal here. The goal here is to see that you're taking glucose, which is a nutrient that you take into your body, and you're turning it into cellular ATP, or energy that the cell can use in the form of ATP. And that's it for today. Thanks.